So Emily is opening the roundtable.
Okay, thanks, Elisabetta. Yep, I'll introduce the roundtable. So I'm just going to share my screen.
And first, I should thank all the speakers for the second part. It was excellent. Thank you.
Okay, so thanks everyone. So we'd scheduled an hour, but it'll be a bit less than that now, I think, for a roundtable discussion about implementing the next stages of FAANG. So ideally, we'd like this to be a discussion, so participation from the audience and sort of, I guess, questions posed that we can address and then take forwards potentially. So if you could post questions or suggestions into the chat box, that would be great. We had 3 discussion items that we posed, and we were going to give each 20 minutes, but I think it'll be more like 15 minutes now to fit in with the time for the workshop. So the first discussion item I was going to lead, which is how to maintain and harmonize global FAANG collaboration. And then Haijun was going to lead a short discussion on the critical need and gap for FAANG bioinformatics and integrative omics data analysis across cells, tissues, and developmental stages. And then Elisabetta was going to give a very brief summary of the results of the FAANG survey and the next steps based on the results of the survey. And then we'll wrap up the workshop from there. So I'll just go straight into the discussion item I had, which was how we might maintain and harmonize global FAANG collaboration. So at the very beginning of the workshop, which seems like ages ago now, I introduced the concept for the FAANG phase 2 meta infrastructure. And one of the things I wanted to emphasize with the infrastructure would be that there would be a strong component of global collaboration in that, and that would include the Global FAANG Initiative and other initiatives, including AG2PI, AgBiodata, and potentially FarmGTEx, which we heard from Ling Zhao today. So we need to, for any infrastructure to be successful and for FAANG to continue and implement into the next phase, we need to continue global collaboration and harmonize what we're doing. So I'd welcome any comments from anyone on this. I actually can't see the chat. I've just noticed whilst I'm presenting, I think, which is very helpful.
No, the chat is still of the previous questions, I think.
Yeah.
People need to maybe to take a moment of break.
I know. I think everybody's there, especially in Europe. It's now.
Yeah, it's very late.
10, and here it's 8:45, so it is quite late for some of us, I think. Yeah, I can't see any questions, but
I make the comment maybe. I think that also all the contents of the excellent presentations today, I mean, it shows that there is an incredible pace going on. And so to really keep collaborations between geographical communities that organize in local infrastructures and so on, it's a key for the future. Of course, we have a FAANG data portal, that is a first big achievement, I would say, when it will be really achieved. But if we got to move further from that, I think we should really harmonize as much as possible. Also because we risk to make overlapping things, because the amount of data is becoming huge. Mm-hmm. There is a question from Chris.
I can't see it. Oh,
Chris,
maybe I can.
You can speak out, Chris.
Yeah, you could speak out.
Yeah, that's fine. I just thought we were putting it in a chat, but so people can see the question. You were asking, one question is what could be a mechanism? How could we try to work together? And AG2PI is a initiative to try to advise the USDA on how to potentially fund relatively large projects in plant and animal sciences for genome to phenome. And so that could possibly be an approach we take to apply to them to bring together FarmGTEx, FAANG, AG2PI, AgBiodata. We're already talking to AgBiodata because they have some clear interests in data reuse, for example, with Peter. So so that could be a possibility. That would be a funding opportunity to try to bring people together, or to try to even potentially support someone in terms of administrative work to try to at least create a mechanism for the discussion.
Okay. Yeah, thanks, Chris. I noticed the data discussion that you had last week with AG2PI, the presentations and things, that was funded by a seed funding grant from AG2PI. So I wondered whether there might be a mechanism to have a similar workshop about harmonizing through the seed fund.
Yeah, exactly. Yeah,
Yeah.
and I think that would be, there's you can go on the ag2pi.org website. I'll put something into the chat that would take you directly to the seed grants. You have to have a PI from within the US because it's USDA funding, but money can go outside the US. You just need to have clear evidence that the US is involved. But just briefly, there's sort of 2 or 3 different grant funding opportunities. One is up to $15,000 where you can get money to put together a working group. And that might be the most appropriate thing I was just thinking about for that. That would obviously fund primarily things like travel to get together, and maybe other resources, electronically, like GatherTown or some other possible resource that you might have to pay for. There are other ones that are perhaps a little bit more involved, and you can apply for $50,000 to $100,000. That particular opportunity is a little bit more structured and has a lot more sort of requirements for the applications, but the meat of the application is 2 or 3 pages. It's really not very much. That unfortunately is due March 8th, so that's coming up very, very quickly. But there's time to put something together like that.
Okay.
So Krista's made a comment about the challenge for sort of the global FAANG is to be harmonizing across the major funding agencies, so USDA, EU, particularly for larger projects. And I think this was particularly with the Horizon FAANG funding that came about. It was partly the drive that had happened the year before in the US that really enabled such a large fund to be put together. So I think there is a strong need, and what we have in the past been quite good at is involving people from those funding agencies to join our discussions. And I think that's an important thing to continue to do. And obviously AG2PI is moving towards a much larger yearly investment of funding on the US side. And really this is partly why we're trying to pursue this infrastructure, is to then try and then be able to hang off national and other EU funding on our side to support the research side that builds around the infrastructure. So I think that is a big part of and Krista's comment, which I think I would echo completely, is that we need to ensure that these actions then lead towards new rounds of funding that actually fund the next phases of the FAANG work, particularly around the things that we've outlined in the FAANG thought paper.
I would second the cross-agency aspect of it. I mean, people were quite willing to talk, and I think that did lead to some real outcomes from we got the funding agency's attention in 2015.
Yeah, when we did meet with them, didn't we, before the FAANG to Fort manuscript as well, where the US funding agencies were certainly engaged with it at that stage. So maybe it's time for similar meetings again.
And the National Science Foundation has funded a discussion group, an RCN, for Plant Cell Atlas. So that's another fairly substantial group. They have a fairly detailed white paper out in eLife last year. And so we're talking with them as well to see, because AG2PI involves plants as well. So, but what we're hoping is that there may be a cyber infrastructure that people would fund, and there are programs both at the USDA and NSF for cyber infrastructure, as I'm sure there are in the EU.
Okay, thank you. I wonder if we continue with this or we move potentially on to Haijun's one, given the time. I'm not sure what Elizabeth or you think.
Maybe, yeah, because
Yeah.
people will start leaving soon. So
I know.
if you feel
It's
that we clarify the points, we can move.
Yeah, no, they're good points. Thank you. No, that was useful, and I shall take those forward. Great. Okay, so I'm going to stop sharing and then hand across to Haijun.
Yeah, I mean, so basically, all of you guys see, the community generate a huge number of the data. Data is continued generate and sharing in the public domain. And then I guess, so from, I think currently from both Europe and US try to think about how we can have build up the bioinformatic infrastructure, to meet the need of data generated, and also have the metadata available, and have a resource available for the scientific community so people can able to utilize the data. And second aspect is, how to integrate all of those data. This is the heterogeneity of the data, even from same type of data, probably have a huge of the difference. How we can, as a community can develop a certain of the resource, share resource, so that people don't have to start from the beginning, right? So we can utilize the available pipeline and, develop, optimize those pipeline, can share it with the community, and then in across between different type of omic data and doing the comparative analysis, and also use these functional data, just Rido mentioned, to the genomic connection, that type of, so I guess for the community, what is the current gap and the need? How we can collaborate together? What kind of a mechanism might be appropriate at this stage for the community? So I think we're open for any questions. Let's see. I see have. Seems like I have a many question here. Right? Oh, this is the
last one, which is a question about if there will be a special session dedicated to FAANG projects at the WC Galp this year. I don't know if it is in line with your.
Does anybody know?
I know.
I'm not able to attend, so I didn't.
It's not a dedicated session. There are talks mostly from Eurofund projects, and they are labeled the same, but and then actually Eurofund also is sponsorizing one of the sessions on functional annotation. You can look at the program when it will be finalized, and you will see.
But I suppose at the VC Galp you can organize something if you want. In that sense, because it may not be part of the VC Galp itself, but there are opportunity to, let's say, rent a room and have a discussion among people that are present at VC Galp about FAANG.
Yeah.
So.
Haijun, there is Ted who would like to ask a question directly. It's your decision.
Who asked a question directly?
Ted would like to ask a question.
Yeah. Go ahead.
Yep. So I don't know how many people on the panel were at the AG2PI discussion a week or so ago, but there were some wonderful tools that had been developed for the ENCODE project, that one would think we could take our data directly as it is, and retool and use within those tools, things that assess the impact of polymorphisms on regulatory, things of that nature. I mean, tools that we simply don't have right now. I mean, so are we, or should we be looking to do exactly that?
I mean, that's exactly, I think as a committee, I think because that's how, when we have so much of the functional data, right? How the functional data can tie it to the SNPs causative variants basically, and then connected to the phenotype, the complex traits. I think that's exactly as a community, we should think about how we can, have certain of the mechanisms through any funding, to develop such a resource for the community. So the people work on the different species can benefit from that.
Okay. I mean, so should we reach out to them and ask, what are the next steps that we need to take to be able to leverage the platforms that you have developed to do what we need?
Yeah, certainly. Yeah.
Okay. I don't know who would do that, but
Yeah.
yeah.
I was working a little bit with some of the human cell atlas people on the single cell, and they indicated to me that that's a huge project for them, that there's 200 bioinformaticists and coders behind the Broad tools to work on single cell RNA-seq. So these are huge projects, but certainly maybe what we can do is find out what do we need to do to hitchhike on the stream, right? And I think one of the biggest questions would be, for me, it sounds to me like you were working in that area, Ted, of thinking about what type of file, what file system and what file type do they need to have the input to all these analysis tools, right? And to me, it seems like that is the important thing. What does the Human ENCODE project do? What does the other projects do? What kind of files do they use and modify and store so that somebody can pick it up and do all these different analyses? And I'm hopeful that you're right, that there is a fairly common file system, a file type that people use, but I'm afraid that there may be a lot of flavors of things. But
I mean, that's probably right. And I would suspect that most of those developers are tool builders, and not, they will have built a database, they will have built the software on top of it, but the work to actually put the data into the places that it needs to be rendered and used by their tools would be a much lighter lift, and something that we could probably apply for modest funding to get done. That's just a guess.
Yeah, I agree with that.
But I think one of the biggest challenges is that people tend to use their own pipelines and their own settings and all these kind of things. So even if you have a bunch of bed files or something else that you provide, and you want to compare across species, there may be underlying differences in how the first analysis has been done. And I think that's one of the biggest challenges. And I think we have tried a little bit in the Horizon 2020 to say, okay, let's try to keep to a little bit these NF core pipelines and to have a kind of standardized thing. But just to cite Alan Archibald, he once said that bioinformatics will prefer to share the toothbrush than to use other pipeline of someone else. So this is really a challenge to get bioinformatics people I don't consider myself as a bioinformatics, so I can say it, to accept other pipelines or other way of analyzing data.
But in the case of an F-core, it was quite successful, not completely, but quite good.
Yeah, but there are probably still people, I know that there are people that are, well, having some doubt about whether they want to improve the NF core, so, but that's fine. I think it's fine. So as long as it is open accessible and everything is put on a public repository, then it's fine to change things. But I think this is, I could imagine, and maybe that's something that Elizabeth is coming to, that if you want to do this, there should be a kind of more global work group that try to discuss these kind of issues together. So from the US, from Australia, New Zealand, China, or Europe, if you want to combine these kind of data.
I mean, we have this bioinformatics subcommittee, right? So I guess so it's a little bit challenging, I guess. We have divided the subcommittee into the different type of the assay for developing the pipeline. But I guess because everyone is busy and then needs not of a huge effort, not just a minor effort. That might be hard to put these things together. So it might be if there's any funding mechanism, that might be can tie the people together, right? To leading this global effort to develop the bioinformatics resource, by collaborating with probably biomedical field, as we know, many of the pipeline from biomedical work in human mouse, not necessarily work in farm animal because they're the structure of the population, and then they're different, and so I think there might be some of the area, as a community, we either through the US or Europe, kind of put on those probably grant together, could be one resource. If you have a grant, funded a grant, and then so people can commit their time and effort, and then we'll have targeted of the objective and a goal to achieve.
There is a comment from Fiona. She says it is important to have specific pipelines, and that does not stop people from having or running their own pipelines. Standard pipelines and workflows will allow benchmarking. So it's a comment. I don't know if Fiona, you want to add more, maybe online, in person?
Well,
Chris agrees.
So anyone, if you want to speak out, please raise your hand. I will able to unmute you so you can speak out. Okay. And anyone, even you are not to the panelists, if you want to speak, please raise your hand so I can see it and unmute you so you can speak.
Okay. Peter, you can speak anytime you want.
I was just didn't want to jump in. So I mean, I think on this point, I think we have been talking about this a number of times. There was a really good talk that I gave on behalf of Mick Watson a couple of years ago from FAANG on sort of his vision for how bioinformatics and FAANG should be done. I think within that and what we've been trying to work towards, there's multiple levels of reusability. So the first stage that we're currently kind of hitting is the ability to capture what people have done with their data analysis. You can openly log against your FAANG data sets which pipeline you've used and how you've used it. And the one that we're trying to move into now is to encourage people to work on the same pipelines and then collectively work on those and utilize those. Then I think there's going to be a third step, which is actually providing cloud-based analysis platforms that can easily get access to the FAANG data and run these standardized pipelines, modifications of those standardized pipelines in either IPython notebooks or in ability to use cloud platforms that then allow groups to then utilize FAANG data without having massive amounts of their own resources. So that's sort of, in my mind, the three stages that FAANG needs to move towards, and there's a huge complexity to each of those stages. But I think we're just now starting to approach that stage 2 with things like NF Core and the ability for people to reuse other groups' FAANG pipelines. But really, I think we need to solidify that and make that common practice, and then start to think about how to move towards this next stage, which is to actually enable people to use these pipelines and use all of the data more easily.
Okay. We see the people volunteer to leading the discussion on that part.
But do you think, Wei Jun, you want to continue this part? Because this links also to the final one, if you want. Yeah. Or you want to keep on?
No, I mean, it seems like I didn't see any other additional comments on question for that. So I think we discussed some of the aspects of potential mechanism, and people can continue for this, but in terms of the time, you know.
Yeah. Time is
you're right. Yeah.
So if you want, I can move on. So the last part is more relaxing, guys. There is nothing so to put our brains in boiling. So can you see my screen?
Yes.
So I think all of you have been invited to this survey early January. It closed in February 8th. And the general idea was to remap the FAANG community where we were, and possibly try also to attract some new people. So I will go very fast, and the aims are reminded in the text box. And in red, I put that, of course, the idea was the survey would provide the basis, bottom-up suggestions to organize new thematic task forces to replace or update the current FAANG working groups. And we had 88 responders, which was not bad, I think, and at least 17 new people who were willing to join FAANG. Because on the left, you see the geographical distribution of these responders. On the right, I asked if they were already a FAANG member, and 17 said, not yet, but I intend to join. So this was encouraging, in my opinion. We also obtained, because this was done together with the FAANG steering committee, other answers on other questions. I'm not going through it because it would be too long. Anyway, I will ask Ziliang and Chris to put this on the FAANG website with all the data in it after we discuss with the steering committee. And when we asked, we proposed a number of items, of areas that could be the future task force, and we allowed multiple option answers. And you can see that what we obtained maybe could be expected, but we have, of course, FAANG Pharma GTEx to study correlations between genotype and tissue-specific gene expression levels in animal population, which is at top. And this followed quite closely by comparative genomics and multi-genome mass visualization and analysis, and FAANG single-cell atlases for animal tissue for fine-scale dissection of complex traits, and so on for the other ones, which are overall quite well represented. Few people said that none of these is interesting. And of course, very often the same respondent indicated interest in many proposed task force, like for example, GTEx and FAANG single cell very often go together. And then they indicated other areas. This was more broad as kind of answer, but they would be interested for potential task force or anything else. And here you see that we cover a lot of potential items, not only more methodological, technical, like the previous ones we proposed, but also more about the real fields, like the first one, epigenetic plasticity and adaptation of genome to environment. And we also find in these questions, in this proposition, suggestions to maybe rearrange a bit those task force we proposed, to include a new item, or maybe to make them broader. Like the guy you at the third before last said, repository of bioinformatic pipelines. This could be linked to what we've been discussing just right now. The other items listed could be, again, used either as new task forces or for broadening the ones we propose, or for targeting, for targeted discussion events that we can also think to propose. And then the very important thing was, would you be willing to lead a task force? And that was nice, because there are 33 people who said possibly, but only 30 mail contacts, because it was, we didn't force people to leave their identity and email, so only some people did. So 30 email contacts said possibly yes, and 8 of them are kind of new and could join FAANG. And the yes are 19, of which 16 mail contacts. So we can contact them together with steering after we elaborate more, we discuss about this. And 4 of them intend to join FAANG. So I think this makes, gives the message that there are people who will to meet, to discuss. We also gave instructions to organize this task forces. And also the opportunities that they are around, like in AG2PI and maybe next from Europe, etc., they can be used. I mean, the same people could be actually put in condition to apply for those possibilities. And here, just to complete, I put some possible items to discuss, like, of course, it's very late now. I don't think we will go in each of these proposed task force discussion, but there could be additional suggestions on areas to cover, maybe after we think a bit more also of all the results we heard today. There is a question, of course, to start with the task force we proposed, or and maybe allow people, once they will be, we will meet them, and where we say, yes, I will lead, to be flexible and to maybe rearrange them as they think it is better, or we should really take an active part in this process. I also think that, as in the past, it would be nice to have at least 2 co-coordinators from different geographical locations to really fit this idea that FAANG should be a unique thing at its nucleus for most of us. And of course, it should be, I think I would really like to have new members and young people to take the lead in this. And again, for the ideal frequency of task force meetings, it's to be decided. It can be, I mean, it depends. It depends on how many will be, how much people will be available. And again, it will be all evaluated better next by the steering committee, but has to follow up and get back to the community. And I asked Ziliang and Chris to share a copy of his presentation on the website, the full one, not this one, because I think it could be nice as a reference to have a look, maybe. That's it. So if you have any thoughts about that, or you want to listen to the next developments, there is some.
Thanks to Elisabetta and Peter and others that helped get that survey done. That was, it looks like to be very useful information. So thank you.
Thanks. Yeah, I think it is. It will be useful as, you know, move on and also to grasp some pieces here and there that we can put together. And there is Mazdak, we're saying that a bioinformatics focused subgroup meeting is definitely needed. Lots has happened that can benefit from practitioners' feedback. Fully agree with that. And Amanda Chamberlain, there is a lot of data now, so maybe a large combined data or metadata or meta-analysis would be a good way to get the groups working together and producing high impact papers. Again, fully agree with that. So I think steering committee has a lot of work to do.
Yeah, people that are on the bioinformatics subcommittee, has it been meeting? I'm not a normal member of that group.
I'm not aware any meeting initially. Yeah, in past couple of years.
So, yeah,
We do have a subcommittee, and it was constituted, but we don't know how much it's been meeting. So
I think we should really refresh activities, and I think new people are the key for that.
Yeah.
I just want to say that I was leading the methylation, and the biggest challenge was that it was more spectator, you know. So when you had a meeting, most people were just listening, and no one volunteered to do anything. So it was like talking to, I don't know if that's an English expression, to a deaf ear, you know. So
I can do a post, we say.
Okay. No, so I mean, it was something that slowly died out, you know, in that sense, because there was not really a lot of, there were maybe one or two groups that had some interest. But I think maybe now where we do have much more data, it may be much more relevant to get these bioinformatics subgroups or group or more alive again.
Yes, and also something I didn't mention is that when we asked, do you collaborate with other large projects, infrastructures, we got answers also from consortia that are different, like that work on other species. I mean, so there could be new blood coming in, what I mean. And like also for the comment of Ted about, you know, what has been developed by ENCODE and other consortia. So in that sense, it would be nice to hear new ideas and propositions. And Amanda, no, Peter Harrison commented. Peter, you want to say it yourself?
Oh, and I think others actually said the same thing as me at the same time. So I think this was echoed by Amanda and Krista and Pierre and others at the same time. There was more, I think a lot of the subcommittee meetings, particularly around bioinformatics at the time, particularly in Europe, the funded projects hadn't really got off the ground at that moment. And although it would have been great to have been able to plan and to plan ahead for some of the pipelines that needed to be developed, whilst people didn't have the data and whilst people weren't actively funded, it was a lot of people contributing time without funding. A lot of the groups were struggling for input, but I think we're in a very different position now. Unfortunately, we probably needed to do this whilst the pandemic was on, but that's made a lot of efforts rather strained on people's time and people's efforts. But I think in many ways, I think if we almost wipe, reset, and then relaunch membership for each of the working groups from scratch, have people re-sign up to them, have new leadership, I think there are lots of larger groups and then lots of really small, very targeted groups that could be formed to solve key questions.
And Emily, you can say that.
Yeah, sure. So when I first was sort of joined FAANG as an early career researcher, the subcommittees were really the way that I became involved. So they were quite active in those days, especially the metadata one. So I think it is a really useful route for early career researchers to become part of FAANG and take everything forwards, I guess. You know, so I think reviving them is a great thing from that perspective.
Yeah. And maybe we can use it's true. I mean, we have the tool of virtual conferencing that has become very, you know, natural So we can really benefit from that maybe more than before.
Yeah, that's certainly true. We used to all have our videos off and didn't interact, but now I think it'll, yeah, much easier.
So, I mean, we will tell you more about this survey and about steering propositions as soon as possible. So maybe we can stop here for that part. Emily, if you.
Yeah, no, yeah, I think we can end the roundtable discussion, and there's, I think, lots of points for us to address from there, particularly on harmonizing bioinformatics and then also re-establishing the task forces. And we'll meet as the steering committee and then feedback to everybody to ensure that, you know, all of the discussions as open as it can be on this. So that just leaves us to close the meeting, I guess. Do you want, Chris and Elizabeth, if you want to do that?
No, why you made the big work.
Sorry, I didn't do that.
So definitely we thank all the outstanding speakers and poster presenters and obviously the organizer, you know, helped to organize this workshop, particularly all the people stay very late in Europe. You know, we really appreciate your passion on the FAANG community. So your inputs and comments always welcome to send to the FAANG steering committee. We were, as all of you guys see, this is a critical stage. So we are looking for the future phase, how we can, you know, basically have a more of international collaboration, you know, with several major big projects, but at the same time, there's many other group of work on the different type of the FAANG project can be utilize the information available for the scientific community. So we looking forward your input and move to the next phase of the FAANG. So hopefully will be a more successful, fruitful for the future research work.
Wai-Jun, can I ask something? So this has been recorded. Is the plan to put the recording on the FAANG.org website, or where do you want to put it?
Yeah, that's the plan. That's something I want to say that for the people who so missed some part of these, so you will have opportunity to review the video. So will be posted on the FAANG.org.
I would just reiterate my thanks to you and to Zhiliang and to the committee that organized this. I think was very thoughtful and well organized. We're actually on time, right? After 6 hours. So I think that's an achievement in itself. And I agree. Yeah, the next 6 months to 2 years will be crucial for, you know, how we make this usable data for people.
I also thank my colleagues and Zhiliang, and that's great. It was a great workshop.
Okay. Well, so you guys have a good day, good evening, good night. Okay.
Good night for me.
Thank you, everyone. Thanks a lot.
Okay.
Thanks, Zhiliang. Thanks, Elizabeth.
Thanks, everybody.
Thanks, everyone.
Bye-bye.
Bye.
Thanks everyone.